Well, I haven't done the gel or anything yet. So I made the gel and I pulled it out for some weeks.
Hi everyone. We're just going to start today. My name is Ryan Sawitsky. For those of you who don't know me, I'm a graduate student from the nutrition department. I'm going to be introducing our speaker. She's actually the first of our semester and also a student here in the grad department. She's in the candidacy of her PhD, also in nutrition. She's been working on this since 2010 and it's finally close to being done. I say finally close, but judging from her CV, she's kind of made the most of her time here. Honestly, just met her the other day, but sorry, I've been looking at your stuff. You're a pretty interesting person. I don't even know if you realize that you're a student still. Since 2010, she's accumulated 11 publications, 2 of which she's the first author and 1 as a co-author. 7 published abstracts presented at a conference, 2 as first author, and 5 abstract poster presentations, with 4 of them her as the first author. But let's also not, overlook the fact that she just recently completed her dietetic internship as well. It's kind of a lot to do. Her publications have been featured in journals such as the Journal of Biological Chemistry, Journal of Nutritional Biochem, and the PLOS1. She's won many competitive academic awards and educational scholarships for what I feel are pretty obvious reasons, and her work experience as a researcher as well as a teaching assistant is quite extensive. She completed her bachelor's degree in China through the Department of Microbiology and Immunology, after which she was a dietetic intern the first time over in China as well, before coming here and having to do it again. She apparently very fond of dance. Talented ping pong, a psychology enthusiast, which was one of my favorite things. She founded a psychological journal and club in her undergrad. Is an avid climber, having conquered climbing almost 3,000 meters, and was also an amateur rugby player. And she was also an athletic escort in 2007 for the Junior World Championships in wrestling, and the test event for the 2008 Olympic Games that were in Beijing, also for wrestling. I had to narrow that down. That was difficult. Please help me in welcoming our speaker, Jim.
Thank you, Ryan, for your introduction. I didn't mean to make you feel confused to sending you the resume. It is a little bit long, but I enjoy to see my previous experience, so I always put everything there. Okay, thank you all for coming. And today I will present the major findings from my research work. Hopefully you guys will enjoy it and we can have a good discussion after this. Let me try first. Okay, so this presentation will include 4 parts: background, chapter 1, circadian control of macrophage activation, and chapter 2, metabolic control of macrophage activation, and then summary. So first of all, as we all know, the obesity has been a more and more critical issue in the United States. So the CDC from 2013 to 2014 has showed that there are more than 1/3 of adults who are obese, and around 71% of adults, they are overweight. And from the WHO data in 2014, we see there are 39% of adults who are overweight and 13% who are obese. So it seems like the obesity is now a global epidemic. So there are a lot of factors contributing to obesity, such as physical inactivity, unhealthy nutrition, genetic and environmental factors. And in the process, we almost always see there's chronic inflammation in obesity. So in recent decades, there are a lot of research evidence showing that chronic inflammation has been closely related to CVD, cancer, autoimmune disorders, bone, joint, muscular diseases, pulmonary disease, neurological diseases, and metabolic disorders. And macrophage plays a very important role in the process of chronic inflammation. So macrophage originates from bone marrow stem cells and then differentiate to monocytes circulating in the blood. When the monocyte infiltrate into some specific tissue, for example, my interest is adipose tissue here, so then it will be activated to macrophages. So macrophage activation actually plays a very crucial role in adipose tissue inflammation and insulin resistance. Actually, there are diverse phenotypes of macrophage. The two common ones are classical activated M1 macrophages and alternatively activated M2 macrophages. The M1 macrophage usually can be driven by NF-N gamma, LPS, or TNF-alpha, and then secrete some pro-inflammatory cytokines such as TNF-alpha, IL-6, and IL-1 beta. And this kind of M1 macrophages usually plays a defensive role because it works like a microbicidal effect. It plays some defensive role in acute inflammatory responses. However, prolonged and chronic inflammation can be pathogenic. Then we need the more anti-inflammatory macrophages, which is M2, to counteract. So the M2 macrophages usually can be induced by IL-4, and it will work to repair the tissue damage and try to protect the surrounding tissues from the detrimental macrophage immune response. So if we can modulate the macrophage activation status, seems like we can control the chronic inflammation so as to control the systemic insulin resistance. Then my research basically want to try to figure out if there are some modulator of macrophage activation. The first part is try to investigate the circadian control of macrophage activation. So when we talk about circadian, first of all, let's think about our daily life. We get up in the morning, go to bed at night. And during the day, at some specific time, we always feel hungry and we have to eat for breakfast, lunch, and dinner. And from the physiological aspect, our body, the hormones in our body also oscillates like around the 24-hour period. And there are always some times, some specific time, our body temperature is lowest and our body blood pressure is highest. And also some specific time, our physical performance can be the best. So everything repeats like 24-hour period. So there seems like a clock in our body. So what's circadian? It is any biological process that displays an endogenous and entrainable oscillation of about 24 hours. And these 24-hour rhythms are driven by circadian clock, and they have been widely observed in plants, animals, fungi, and cyanobacteria. All the circadian rhythms are endogenous. They are adjusted to the local environment by external cues such as light and temperature. So this core loop of circadian clocks is very important and is responsible for rhythmic biological processes. So the period 1, 2, and 3, and the correct 1 and 2, they are very important clock genes. their transcription and translation is rhythmic. So when after the expression, the product per 1, 2, 3, and the cry 1, 2, they will form a complex which will go back to the nucleus to inhibit the expression. So this feedback loop actually can control the whole transcription process, and also will control the rhythmic of other circadian control genes, we call it CCGs. So the whole process usually takes 24 hours. What we have learned about circadian and inflammation, some evidence already show that circadian clocks in peripheral tissues and cells drive daily rhythms and coordinate many physiological processes, including inflammation and the metabolism. And inflammation or inflammatory responses are rhythmic, and macrophage also has rhythm. An interesting one is half-a-day-induced adipose tissue circadian clock dysregulation in conjunction with adipose tissue inflammation. So the mice, when we have that, then the circadian dysregulated. So my hypothesis is that circadian clock dysregulation enhances macrophage pro-inflammatory activation in diet-induced obesity, resulting in adipose tissue inflammation and systemic insulin resistance. I will present major data for this study, and it includes 3 sections. The first section, I will run in vivo, in vitro study to test if high-fat feeding induces circadian clock dysregulation, specifically in macrophages, including adipose tissue macrophages and the bone marrow-derived macrophages. And the second one, I want to test if disruption of the clock genes period 1 and period 2 exacerbates the macrophage pro-inflammatory activation in vitro. And third, I want to test if macrophage-specific disruption of per1, per2 exacerbates diet-induced inflammation and insulin resistance in vivo. So the first question for the first part is that, is there existing macrophage circadian dysregulation in diet-induced obesity? So to test this, I used the animal model, which is developed and widely used by the scientists who is in the circadian field to address the possible link between metabolic phenotype and the alterations in circadian clock function. The homozygous M2 loop knock-in mice was used in this study. Basically, it inserts one luciferase at the downstream of mouse period 2 gene. Why they choose this gene? Because this gene, the circadian rhythm of this gene, of the mRNA or protein is very robust. That's why they want to insert this luciferase downstream so that we can see the circadian pattern. So the product period 2 luciferase fusion protein can be used as a real-time reporter of circadian dynamics. Because these mice, their background is C57BL6J, so they can still be considered as wild-type mice, because after the insertion of this luciferase, other phenotype didn't change. Still, we can still consider as wild-type mice. So we feed these mice with high-fat diet for 12 weeks, and the control group we feed low-fat diet for 12 weeks. And then, as consistent with all the previous findings in the wild-type, in the high-fat diet-fed wild-type mice, we observed the increased adiposity and the systemic insulin resistance in the high-fat diet-fed group. And from the well-running activity, we can see the low-fat diet group still maintain about 24-hour circadian when exposure to the constant darkness. But the high-fat diet feeding group, the period is prolonged. So it tells us the high-fat diet fed M2 look mice exhibit an increase in the period of the circadian rhythm. And then I want to look specifically in the macrophages if the circadian rhythm has been dysregulated or not. Here I use 2 kinds of macrophages. From the panel A, it is the macrophage isolated from adipose tissue. We use SVC, which is strombovascular cells. So the main population is macrophages. And we can see the period So the red color, high-fat feeding, after high-fat feeding, the period is prolonged, and the amplitude is also weakened. And consistent with this data, these macrophages, bone marrow-derived macrophages, we see the similar pattern. The circadian is dysregulated in the macrophages. And I specifically look at the inflammatory response in the bone marrow-derived macrophages. We can see here the P54 and P46, they are junk phosphorylation, and P65, they are NF-kappa B phosphorylation. These two phosphorylation is a very important indicator of pro-inflammatory responses in the signaling cascade. So after high-fat feeding, we see the increase of phosphorylation of both junk and NF-kappa B. And the MR level of some pro-inflammatory cytokines, such as IL-1 beta, IL-6, and TNF-alpha, also increased significantly compared to the low-fat diet group. So the conclusion for this section is that high-fat feeding induces obesity, global circadian dysregulation, and also specifically induce macrophage circadian dysregulation together with macrophage pro-inflammatory responses. So the next question is, does macrophage circadian clock dysregulation contributes to macrophage pro-inflammatory activation? So I ran the in vitro study. I basically isolated the bone marrow-derived macrophages from the PER1, PER2 knockout mice. Because previous study has found that when PER1, PER2 double knockout, the circadian rhythm will be dysregulated. That's why I choose these mice, trying to intentionally to get the circadian dysregulated. Then I want to investigate if macrophage the activation is influenced by the disruption of circadian. So this is the method of the isolated bone marrows from the leg, and then different plate on the cell culture plates and differentiate to bone marrow derived macrophages. Now I run a flow cytometry to test the population of macrophages. There's one figure I didn't show here. Is first I gauged the mature macrophage cell surface marker F480 and CD11B, and I found the double positive. They are mature macrophages. Among that population, I gauged the CD11C and the CD206 because these two are cell surface marker. Okay? Because these two are cell surface marker for the CD206 is cell surface marker for the M2 macrophage, and CD11C is a cell surface marker for the M1 macrophages. So I want to see how much or how many macrophages are M1 activated and how many are M2 activated. So from panel B, we can see there's more M1 activated macrophages in the PER1, PER2 disrupted macrophages. And M2 macrophages, there's no significant difference. So the disruption of clock gene in the macrophages can exacerbate the macrophage pro-inflammatory activation. And then I test the pro-inflammatory responses by doing the Western blot. We can see there's increased phosphorylation of junk and NF-kappa B in PER1, PER2 disrupted macrophages. And also consistently we see increased pro-inflammatory cytokines such as IL-1 beta, IL-6, and TNF-alpha in the PER1, PER2 disrupted macrophages. So the conclusion for this section is that PER1, 2 disruption exacerbates macrophage pro-inflammatory or M1 activation. Then the next question is, so is macrophage circadian clock dysregulation only will be sufficient enough to bring about a systemic insulin resistance? Then I try to use a bone marrow transplantation to form a myeloid cell-specific disruption of PER1, PER2 animals. So the donor is PER1, PER2 double knockout mice or wild-type mice. Then after get the bone marrow cells from the donor transplanted to the irradiated wild-type mice, which is recipients, and then form a chimeric mouse. And after 1 month of recovery, I feed the mouse with half a diet for 12 weeks. So there are two groups. One group is BMT wild type. The other group is BMT per1 per2 knockout. So first of all, I try to confirm if the transplantation is successful. So I use the primer of per1 per2 mutant and also the per1 per2 wild type to see if the expression if the expression of the mutant is existed. From the A and B, we can see the bone marrow cells and the BMDM only mainly have the per1 per2 mutant band alleles, and because the stromal vascular cells and adipose tissue, they have some other cells together with macrophages. So we can see there are two alleles, wild type and mutant alleles. So that means the transplantation is successful. Okay, and then after half-diet feeding, I observed that in the BMT per1 per2 disrupted mice, we see the a significant increase of body weight compared to the control group, and a significant increase of adiposity compared to the control group. Also, from the F4 staining, we can see the adipocyte size. The size of the adipocyte is bigger compared to the control group. Meanwhile, there are more this kind of crown-like structure, which shows the macrophage infiltration in the adipose tissue. And systemically, we see the significantly higher plasma insulin and also glucose levels in the BMT per1 per2 disrupted mice, and also the insulin resistance and glucose intolerance in the BMT per1 per2 disrupted mice compared to the control group. So then I tested the adipose tissue inflammation. Clearly, we can see there's more MCRB and junk phosphorylation in the BMT per1 per2 disrupted mice compared to the control group. And also, we see the MR level of pro-inflammatory cytokines such as IL-1 beta, IL-6, TNF-alpha also increased in the adipose tissue. So the myeloid cell-specific per1 per2 disruption exacerbates adipose tissue inflammation in obesity. Now I run the flow cytometry FACS to double-check the population of the macrophages in the adipose tissue. So from here, F480 and CD11B double positive population, this is the mature macrophages. Apparently, the BMT per1 per2 disrupted mice has much more mature macrophages in the adipose tissue. And there are significantly higher M1 populations in the mature macrophages. So the myeloid cell-specific per1 per2 disruption exacerbates mature macrophage infiltration in the adipose tissue and also macrophage pro-inflammatory activation. And the insulin signaling in the adipose tissue is apparently impaired in the BMT per1 per2 disrupted mice. We can see the phosphorylation of AKT is significantly lower compared to the control group. And also there are some important genes in the adipose tissue. The MR level is abnormal. For example, the adiponectin and is lower than the control group, and the resistance is increased compared to the control group, and the hormone-sensitive lipase also decreased. So the conclusion for this section is that per1 per2 disruption specifically in macrophages exacerbates systemic insulin resistance and adipose tissue inflammation in diet-induced obesity. Meanwhile, it can exacerbate mature macrophage infiltration and macrophage pro-inflammatory activation. So the summary for the first part is that macrophage circadian clock dysregulation is a key process in the physiological cascade by which diet-induced obesity triggers macrophage pro-inflammatory activation, adipose tissue inflammation, and insulin resistance. So now, even though it's still early to generate some recommendation for humans because it's a mice study, but at least it gives us some ideas. The circadian is very important to modulate macrophage activation and also inflammation. Then next, I want to investigate if there are some metabolic control of macrophage activation. Then I choose this gene, PFKB3, which encodes IPFK2, not only because PFKB3 is one of the circadian control genes, but also because PFKB3 is a very important gene in the metabolic pathway glycolysis. So, as we are all familiar, in the glycolysis pathway, 6-PFK1 is very powerful and the key rate-limiting enzyme in the process. And PFKB3 encodes the IPFK2, which the enzyme catalyzes this reaction, generate fructose 2,6-bisphosphate, which is a very powerful activator of this gene. Then that means the PFKB3 will help with the glycolysis. It will promote glycolysis. And from the previous findings in my lab, they have done a lot of research, either PFKB3 disruption in animal model or PFKB3 overexpression in animal models. They found that PFKB3 protects against diet-induced insulin resistance and pro-inflammatory responses. So I was wondering if PFKB3 can be a metabolic modulator of macrophage activation. So my hypothesis is that PFKB3 disruption enhances macrophage pro-inflammatory activation in diet-induced obesity, resulting in local and systemic insulin resistance. This study also divided to 3 parts. The strategy is very similar to the last one. So the first one, I want to test if disruption of the PFKB3 exacerbates macrophage pro-inflammatory activation in vitro. And then I want to do some in vivo study to test if myeloid cell-specific disruption of PFKFB3 exacerbates diet-induced inflammation and insulin resistance. And third, I want to test if PFKB3 disrupting macrophages has some direct effect on the adipocytes to exacerbate adipocytes inflammatory responses and insulin resistance. So first study, I isolated the bone marrow cells and differentiate to BMDM. Try to use the PFKB3 disrupted macrophages to run the in vitro study. So we can see when PFKB3 disrupted, when the bone marrow cells isolated from the PFKB3 disrupted mice, the expression of PFKB3 is decreased. And then the glycolysis in the PFKB3 disrupted macrophages also decreased significantly. And then the MCRB phosphorylation is increased in the PFKB3 disrupted macrophages. And the pro-inflammatory cytokines such as TNF-alpha and IL-6 also significantly increased in the PFKB3 disrupted macrophages. So from this slide, we can see that PFKB3 disruption exacerbates macrophage pro-inflammatory responses. And then I used IL-4 to try to induce the macrophage M2 activation. Want to see if PFKB3 disruption will have some effect on the macrophage M2 activation. We can see that. Let's look at the black bar first, which is the control group. We can see when IL-4 treatment, the pro-inflammatory cytokines such as IL-6 and IL-1 beta all decreased when IL-4 treatment. And then we look at the white bar, which is the PFKB3 disrupted macrophages. We didn't see that patent effect of the IL-4 treatment. So it seems like PFKB3 disruption suppresses anti-inflammatory activation of macrophages. The panel A is from the bone marrow-derived macrophages, and panel B is from peritoneal macrophages, which show the similar pattern as the panel A. So the conclusion is that PFKB3 disruption exacerbates macrophage pro-inflammatory or M1 activation and suppresses macrophage M2 anti-inflammatory activation. Then the next question is that if the macrophage PFKB3 disruption alone will be sufficient enough to bring about systemic insulin resistance. So I also use the bone marrow transplantation method to form the myeloid cell-specific disruption of PFKB3 mice. And I want to test if it will has the increased diet-induced inflammation and insulin resistance. So similarly, donor can be wild type or PFKB3 disrupted mice. The recipient is always wild type. And then we, from the body weight data, we see from two groups there's no significant difference between them in the body weight or adiposity. However, we see significant difference in the insulin and glucose tolerance test. So we see in the myeloid cell-specific PFKB3 disrupted mice, there's severe insulin resistance and glucose intolerance, and also there's significantly higher plasma glucose and the insulin level. And from the F480 staining, is the PFKB3, the myeloid cell-specific PFKB3 disrupted mice, we see there are more crown-like structures. It gives us the first impression that there are more macrophage infiltration in the adipose tissue. And then I test the adipose tissue inflammation. We see higher, significantly higher phosphorylation of NF-κB. And also the insulin sensitivity in the BMT PFKB3 disrupted mice is impaired compared to the control group. So myeloid cell-specific PFKB3 disruption exacerbates adipose tissue inflammation and insulin resistance. So the conclusion for this section is that PFKB3 disruption specifically in macrophages exacerbates systemic insulin resistance and adipose tissue inflammation in diet-induced obesity. Then next question is that if PFKB3 disrupted macrophage has a direct effect on adipocytes inflammatory in vitro. So I ran a co-culture study to put the wild type versus PFKB3 disrupted macrophages together with the adipocytes cell lines, 33L1 cells. And then I see the when the adipocytes co-cultured with PFKB3 disrupted macrophages, the phosphorylation of NF-κB and the junk all increased significantly, and the insulin signaling is impaired in this adipocytes. So it tells us PFKB3 disruption in macrophage exacerbates adipocyte inflammatory responses and the insulin resistance. So the summary for this second study is that PFKB3 regulates macrophage activation so as to protect against diet-induced insulin resistance and inflammation. So in conclusion, from these two studies, I didn't look at the physical inactivity. I used a Harvard diet to induce the obesity, or some genetic factors such as disruption of some genes, circadian or metabolic related, and then to modulate the macrophage activation. In the process, we see it's very important for the obesity and also insulin resistance. I think that's my last slide. And last but not least, I want to thank my advisors, committee members, and collaborators, Dr. Wu, Dr. Smith, Dr. Alanis, Dr. Wozam, Dr. Cole, Dr. McMurray, and Dr. Ernest. Thank them for their guidance and support. And also all my lovely members in the lab, Li, Cindy, Wu, Ya, Dr. Liu, and Lu, and Jing. And thank you all.
So plenty of time for questions? Questions? Yes.
Any questions?
Oh, okay. I shall give a student chat first. But anyway, well, this is an excellent presentation. I really enjoyed it. You present very convincing. I think you paint a very convincing picture about the macrophage effect in relate to circadian impact, and also show the data about crosstalk impact on adipose inflammation, the whole body insulin resistance. I'm fully convinced that, but I just want to challenge you to think in terms of the data you see, like your ITT, you see this insulin resistance change very quickly, like in 30 minutes you see a, like quickly drop. As you know, the insulin resistance in that kind of short time is the muscle contribute to the whole body insulin resistance. In any of your system, have you looked into the muscle contribution?
I didn't look at the muscle contribution in this study. My main interest is the adipose tissue, but I did look at the liver tissue. Liver display the similar pattern as the adipose tissue. It has more fat deposition in the liver and also more inflammation. But muscle, I didn't look at muscle. That's a very good question.
Because you have very impressive data, that.
Yeah. For the circadian part, maybe I can look at muscle to see if muscle can be one contributor for the whole systemic insulin resistance. But for the PFKB3 study, because that gene only expressed in adipose tissue macrophages, so maybe I'll try to find some other isoform to see the muscle effect.
Jim?
Did you look at mitochondria lipid handling? The notion, mitochondria lipid handling. The idea is that with reduction in glycolysis, you have kind of a reverse Warburg effect. And if you read some Floyd Demetrius's papers, et cetera, database and hypothesis papers, they suggest a lot of these metabolic disorders are because of a reverse Warburg effect. And on the other hand, increased glycolysis might increase cancer risk. So can you address that?
That's a very good question. Actually, PFKB3 gene, this gene is a very interesting one. I think the philosophy is sometimes if it's too much, it's not good. If it's less, if it's too little, it's also not good. The PFKB3 can be a very good example because our lab already published one paper about PFKB3 in the circadian. So when circadian disrupted, PFKB3 if upregulated, it will result in cancer. So because cancer or maybe use the glycolysis. So, but in my study, I see, because I didn't see the cancer, I only see the insulin resistance. I see PFKB3 is protective. So I mean, for the inflammation or insulin resistance perspective, I want PFKB3 to be there. But maybe for cancer, if we have the cancer stage, maybe PFKB3 expression high, maybe not good.
Were mitochondria enhanced, but in a sense they were dirty mitochondria? Usually you would think mitophagy might be decreased.
I have no idea.
No idea. Say again.
Thank you.